Hi, I'm going to talk to you about development of antibiotic resistance in bacteria and how it's evolved. Okay, so remember, antibiotics are chemicals, drugs that we can take to kill bacteria when they're inside our bodies, discovered by Alexander Fleming, and now we have lots and lots of different types. Now, what we've got to think about is imagine we've got a population of bacteria, and I've drawn it like a petri dish, but imagine we've got a population of bacteria, lots of different of the same type of bacteria, lots of different colonies. Okay, now within these colonies here, we might have some bacteria that are naturally resistant to antibiotics. Let's say the antibiotic is penicillin. Now, if there's no antibiotic being used, they'll be in the mix along with the non-resistant ones or the susceptible ones. Now, that mutation may have occurred just by chance in their genetic material, or they may have picked up an extra gene for resistance on one of those little extra circles of DNA the bacteria have called plasmids. Okay, but if we start using an antibiotic, okay, such as penicillin, or it could be something like streptomycin or erythromycin, whatever it is, okay, we add that into the environment, so maybe we've added it into the agar in our petri dish. What that's going to do is that will kill all the susceptible bacteria. Because they don't have the resistance, so they're all going to be killed off. And what that leaves behind is the resistant ones, because the resistant ones are not killed. And that's really the key, they're not killed. And if they're not killed, that means they can reproduce and pass on their gene for resistance to their offspring. Okay, so they're going to reproduce and pass on the resistance gene. So over time, more and more of the bacteria become resistant. So the number of resistant bacteria in the population increases. Now you can imagine if that's in a patient who's got bacterial disease, they're not going to be able to be treated by that antibiotic anymore. As long as you keep using that antibiotic, it keeps selecting for the organisms that are resistant, that have that adaptation, key term adaptation, that makes that they are resistant because they're not killed, and they reproduce and pass on their gene to their offspring. So over time, more and more of them will be resistant to that antibiotic. Bacterial resistance to antibiotics can evolve really quickly, and has evolved really quickly, because they have such a short generation time. Remember, we talked about the fact that bacteria can reproduce every 30 minutes maybe. So they can go through thousands of generations of bacteria within a few days, even less. So this is why this is evolution in action. This is actually evidence for the process of something called natural selection. Okay. Now in hospitals and in general in medicine, we've used antibiotics sort of almost willy-nilly in the past, and we give it to people who've got viral infections. Remember, antibiotics do not work on viruses. Okay, because remember, they live inside your cells, and they also don't have any of the features that antibiotics actually work on, such as disrupting cell wall synthesis or anything, or protein synthesis or anything like that. So they're not going to be effective, but people do take them when they've got a cold or a mild viral chest infection, throat infection or chest infection, and that just adds to the problem. Also, farmers have used antibiotics in their feed to try and keep their animals healthy and make them grow faster, and that selects for any resistant ones in the food chain, and that means then we can get infected by bacteria that are resistant to antibiotics as well. Okay, and obviously in hospitals they've been used a lot, and this is becoming a real problem. And you might have heard of a disease called MRSA. Okay, and this is called methicillin-resistant Staphylococcus aureus. Okay, so now Staphylococcus aureus is actually a bacteria you probably have on your skin anyway. It causes spots and things like that. But if it gets under your skin through your barriers, it can cause really nasty subcutaneous, i.e. under the skin infections. And if it's resistant to lots of antibiotics, that can be real problems to treat in hospitals. And sometimes it's not just called methicillin resistant, it's called multiply resistant. Yeah, so. Okay, so in other words, multiple resistance. Okay, so it's resistant to lots of different antibiotics, and that means it's very difficult to treat these infections. Simple things like hand washing in between patients to stop the transmission of bacteria from one patient to another has been really fundamental in hospitals. Just looking at some evidence, you might be shown graphs in the exam. Okay, here's a graph looking at levels of antibiotic resistance in a couple of hospitals, and this is actually dates back to the 1950s, so you can see this was becoming a problem even then. So you can see up to 1956 there were no resistant bacteria in either of these hospitals, but they'd started using this antibiotic erythromycin at the same time. And you'll notice that the antibiotic resistant appeared in both hospitals almost about the same time. Okay, so there must have been some sort of mutation occurred or picked up by the bacteria and they started being resistant. And as we use the erythromycin, we're selecting for those resistant bacteria all the time, so they increase in their numbers. And you can see in Hospital W, there's a huge number of the bacteria, about 65% are resistant to antibiotics. Okay, it's lower in Hospital E. Maybe they weren't using the antibiotic as much, so maybe there was less selection going on. All right. Now, key thing here is they stopped using the antibiotic slightly after each other. Okay, Hospital W waited a bit longer. Okay, and that's why they've got a much higher peak, because if you look at it, there's not that much difference there in those two. And almost as soon as they stopped using them, the level of resistance went down, and that's possibly because non-resistant bacteria reproduce ever so slightly faster than resistant ones, because being resistant requires a bit more energy, so it makes you reproduce more slowly, and maybe they outcompeted them, I'm guessing here. Okay, but that's really an important thing. If you stop using the antibiotic, actually you're removing that selection, so those ones no longer have an advantage in the environment, and that's what hospitals need to do. They need to rotate the antibiotic they use, stop using them entirely for a time, to stop this resistance from spreading. So hopefully that makes a little bit of sense to you. Thanks.